Welcome everybody to a StriaTech hosted journal club. My name is Thomas Münch and I'm one of the founders of StriaTech. I welcome you to another of our journal clubs where scientists present their projects and data, highlighting the use and the application of our products. StriaTech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The StriaTech founders are all experienced neuroscientists in the field of vision research. Today, we welcome Reece Merced as our speaker. His talk is entitled Monitoring Refractive Development in Mice to Investigate Mechanisms of Myopia. Reece obtained his PhD in the lab of Erika Eggers at the University of Arizona, where he studied retinal circuitry. During his postdoc as an NIH fellow at the State University of New York, he switched to looking at signaling pathways in the visual cortex. He is now working with Michel Pardue at Emory University School of Medicine, where he applies his expertise to study the mechanism of myopia. Today, Reece will introduce us to some background on myopia and will then show us many examples where the measurement of refractive development in the mouse eye brought about new insights into the mechanisms of myopia. Before you start, Reece, let me remind the audience that you will be able to ask questions throughout the event and also to give a thumbs up to other questions. We will get to those questions at the end of Reece's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right-hand side will stay available for a couple of days after the live event. So if you're currently not watching live, but a recording of the event, and if Q&A is still available, please make sure to enter your question and your name so that we can back to you with the answer. We are very much looking forward to your questions. So Reece, please go ahead with your presentation. We're very much looking forward to it. You can share your screen, unmute your microphone, and yes, please go ahead. Okay, screen is there. Is your microphone on? Can't hear you. Your mic isn't working. There we go.
Okay. Sorry about that.
And you need to share the screen again, please.
Okay, there we go. Can you hear? You can hear me and see the screen.
Yes.
Okay.
Yes.
Okay. Great. Yeah. Well, thank you so much. I'd like to just thank again for the opportunity to talk about some of the work that we're doing in the Pardue laboratory and that has been done in the past. And I'd also like to thank everybody who's tuning in to hear this. And today I will be talking about how we use the photorefractor to monitor refractive development to study myopia. And so first I will define myopia, describe some of the general ideas about factors that may drive its progression and some of the questions that we have. I will then discuss how we use the photorefractor in the laboratory, and then I will present a series of studies from the Pardue lab where we use the photorefractor to investigate mechanisms in the myopic mouse. So myopia, or nearsightedness, is a problem where near objects appear clear, but distant objects appear blurry. And the rates of myopia are rapidly rising around the world, in particular in East Asian locations, as you can see in this graph here, where the prevalence is almost 80%, in 2010 in 20-year-olds. And in fact, it's estimated that almost half the world population will become myopic by year 2050. So this is a really significant public health problem. And while myopia and the blurred vision can be corrected optically, having myopia still increases the risk of developing severe blinding diseases, such as glaucoma, for example. And there's a big economic burden from lost productivity due to vision impairment from any complications. So as light enters the eye, it's refracted by different structures in the eye, and then it will land somewhere on the back of the eye, hopefully to be detected by the retina. And when there's a mismatch between the eye's optics and its structure, this focal point does not land nicely onto the retina, and this creates a refractive error. So here is a graph of a neonatal population, a histogram of a neonatal population with the refractive error in diopters on the x-axis. And you can see that early in childhood, early in development, eyes are slightly hyperopic. So actually the focal point lands behind the retina. And then as development occurs during childhood, the process of emmetropization happens, and when it's successful, all of the optics and structure of the eye line up nicely, and the focal point of visual input lands on the retina, leading to clear vision. And you can see in this graph in red, the population is around 0 diopters, so there's no real refractive error here. However, when this process fails, a failure of this process leads to myopic eyes, as you can see in this graph, where the focal point of visual input actually occurs before the retina, and this leads to visual defocus. And so what defines myopia is really a refractive error, which causes the light that enters the eye to be brought to focus in front of the retina. And then a quantitative component can be added to this definition so that this refractive error needs to be at least 0.5 diopters. However, there are multiple ways to create a myopic refractive errors. One way is called refractive myopia, and this is due to changes in the image forming structures of the eye, like the cornea or the lens. So if you modulate the optics of these structures, you can create a myopic refractive state because the light is focusing in front of the retina. You also can create a myopic refractive state with an increase in axial length of the eye. So this might be a more familiar sort of terminology that people may know, but this is essentially where the eye is too long or too large for its optics, and occurs a lot in human myopia. But this achieves the same problem here, where light is focused in front of the eye, leading to blurred vision. And so regardless of the type of myopia, you really need a change in refractive error or a change in refraction and your measurements of refraction to have a myopic eye. And so a large, sort of the main question in the field is what causes this myopic refractive state to develop? And the short answer is it's still unclear despite this large amount of effort. But the longer answer, slightly longer answer, is that we know there are many potential important players here. And one of the most significant findings in the field, I think, is that spending time outdoors is protective against myopia progression. And there are a lot of different components of outdoor environments versus indoor environments, for example, that could be involved here and that have been studied both in humans and animal models. One is luminance level, so brighter light gives more protection against myopia. Another is the wavelength composition of the light. So indoor environments are deficient in certain wavelengths. I'll talk about both of these things with some studies from the Purdue laboratory later. Also, there's differences in spatial frequency, blur, and a whole host of other things that are represented in this cartoon here. And so I think that it is very important to start identifying mechanisms that underlie myopia progression. And this is especially important because this can help to give the most effective prevention or treatment options. And so in order to study myopia in a mechanistic way, animal models are really critical here. And there have been a number of different animal models that are used in the field and continue to be used in the field. I have some of them listed here. In the Purdue laboratory, we use the mouse model. There are many benefits to using the mouse, which principally include the ability to genetically manipulate the model in a number of different ways. And I'll be talking about some of these, how we utilize this kind of manipulations in the laboratory. And then also there's other things like their visual system is very well characterized. So we know a lot about kind of retinal processing and visual processing and eye structures. But there are challenges, and one of them, one of the main ones, is that it's technically challenging to study such a small eye, and especially in terms of making refractive measurements. But because monitoring refractive development is so critical to studying refractive development and also myopia, some special options had to be made for this. And this was done and accomplished by Professor Frank Schafel, who developed this automated eccentric infrared photorefractor. This image on the right here is a newer iteration of the device currently. And here is a image of, in the top right corner, of sort of one of the original style devices. You can see it consists of a camera that has some LEDs on it. And how this works is that the camera detects the infrared light that is projected back out of the eye, and you get a brightness profile of the mouse eye. So in this image, you can see the mouse head, and then the pupil is nice and large and it's nicely lit up. And then the inset on the left is a brightness profile. And the camera detects this, and then it plots the brightness of the pixels and fits a line to it, which is what the arrow is pointing to. And the refractive error is determined from the slope of the regression through these pixel brightness values down the vertical meridian of the pupil. And then this will give a readout of refractive error in diopters, which is you can see at the bottom here. And so in this particular example from the 2004 paper, from this paper, this reading is about 9 diopters. So this is eye is hyperopic. It has a plus 9 diopters of refractive error, and you can see it has a positive slope. Another thing to note is that hyperopic eyes tend to have brighter pixel values at the top of the pupil and dimmer pixel values at the bottom of the pupil. And there are a couple important parameters here that are included kind of in this calculation. One is a conversion factor, and this is determined through the calibration of the system with a series of trial lenses. And then this recording can be very fast, and it relies on the video frame rate. And here is another example of, instead of a hyperopic eye, a myopic eye. And so hopefully you can appreciate that the pixel brightness of the pupil looks slightly different. Myopic eyes tend to be darker on the top and brighter on the bottom. The slope of the line is now negative, and the refractive error in diopters is minus 11 diopters. The measurements using this infrared photorefractor are fast and also fairly reproducible. So here is a graph showing refractions in the left eye versus the right eye. And if you look at the individual points, you can see they match up quite nicely. But there is a lot of variability here, as you can see with the error bars. And some of this is due to some challenges here. So one is the eye orientation and the alignment. So the eye has to be pointed towards the photorefractor, but you might be able to imagine that small changes in how the eye is aligned could affect this reading, and I will talk a little bit more about that in a second. Another consideration is the small eye artifact. This is a phenomena where light entering the eye of a small eye, like the mouse, actually will focus a little bit anterior to the retina. And so the light that we detect and the readings that we have may be slightly biased towards hyperopia, or maybe not so slightly biased, but just rather biased towards hyperopia. And so this is a consideration as well. And so now I want to kind of walk everyone through how us in the Purdue laboratory do these refractive error measurements. And there's a few important considerations before we start to make sure that are done. One is to have the room be dark. Sometimes we'll use a dim red light, or just even the light from the computer monitor that we're using is enough, but it should be dark in the room. We always will dilate our pupil. In our case, we use often tropicamide, but we want the pupil to be really large so that we get enough brightness to be able to make these accurate measurements. We wanted to make sure that the pupil is bright and round so the dilation works before you start recording. And then it's really important to avoid disrupting the tear film. The tear film can be disrupted even when you think you haven't done anything, like if the animal accidentally hits its eye on the side of the cage or something like this. And this can create a lot of difficulty in getting these refractions, and I can discuss this more if there's interest. And so first we can take awake refraction. So this is just an awake mouse. There's no anesthesia here. And this can be done a couple different ways. Originally it was done by placing a mouse on a platform angled towards the camera and by gently kind of holding the tail. And then to move the eye around, you move the platform around. There's also some methods that involve some light restraint of the animal to make this a little bit easier. This is a difficult kind of technique to master, and it requires, having the mouse feel comfortable so that they're stationary for some time on the platform. But when the eye is angled towards the camera, you get an image that you can see here on the right, and you see that this cyan circle encompasses the nicely bright pupil. This red crosshair is the center of the pupil, and then the circle is the Purkinje image, which is a reflection off the cornea. And so by using the distance between these two values and locations in the image, we can try to get a gaze direction and try to align the eye. I'll talk a little bit more about that later. And then to the right of the pupil, we can see a yellow fit line, which is fitting the pixel brightness values. And so in this particular eye, you can see this line has a really big slope, and so it's very hyperopic. And then the program and the software will give you a few values as well, which you can see in the inset on the right of the image. In purple is the refractive error. So this is, in this particular example, this is a 14 diopter eye, and then it will give you a standard deviation of that error. It will also give you pupil size. We want the pupil to be at least 2 millimeters, so it should be nicely dilated. It also gives you a readout of the pupil pixel brightness, and you want this to be kind of as high as you can sort of get it. And I will talk a little bit more about nuances with that in a second. And then it calculates this gaze direction. So you can see this crosshair and this Purkinje image are not perfectly overlapping, and so there is a discrepancy in degrees of that alignment. And so for awake measurements, as you might imagine, the mouse can move, so it's a little bit more difficult to get a lot of stable measurements. So we will collect at least 10 good measurements, and a good measurement would be the eye is stationary, there's enough alignment here, and then the program will pick that up as a recording. But because there can be variability in all recordings, regardless of how you're doing it, we have some thresholds that we apply. And so to count as a good measurement, we make sure the standard deviation in awake refractions is less than 1 diopter. The gaze, the center of the gaze should be less than 10 degrees. And then because we measure both the right and the left eye per mouse, we also want to make sure that the eyes match, which they should. If they don't, this is another, there can be something else going on, and I'm happy to talk about that as well. But for our sort of thresholds, we make sure that the difference between the eyes is at least less than 2 diopters. And then if this measurement reaches each of these criteria, then it is included. Otherwise, it may trigger some exclusion criteria. Okay, so you can take awake refractions of the animals. You can also now do a more systematic measurement under anesthesia. So the strategy here is that after the mouse goes asleep, you can place them on the platform and then raise the head slightly to a more natural position. This can help with the eye alignment. The first thing we do when we do this is we want to align with the optic nerve by moving and rotating the platform around. And so here's an example of what that looks like. First, on the bottom left image, I have just a retinal fundus photo with some concentric circles. You can see the optic nerve in the middle and then 2 rings around it. This is just kind of a cartoon to help orient you to how sort of the fundal area that we're looking at when we are measuring refractive error, and you'll see this in a second. So first we align on the optic nerve, and when we do that, so we're in this location, when we do that, you can see that the pupil is very dark. It's nicely dilated, but it's a really dark pupil, and we actually get almost like zero. The fit line here is almost no refractive error, or often it will be myopic, and this is something that was noted with the original paper. But this might not be a great location to take a measurement because it's so dark. We're not really getting any sort of pixel changes here. And so then the first thing we do is we rotate the platform slowly in one direction, and we always usually start with a horizontal direction. When you do that, when I do this and show the images, you'll see that the pupil brightness increases, and then it will kind of saturate and it won't get any brighter. And you'll see that there's a better on-axis alignment as we do this. So first we'll move it slightly. You can see that there's not a good alignment, but we start to get a more hyperopic slope, and the pixel brightness starts to get slightly brighter. If we continue, now we're in a position where we have good pixel brightness, we have really great alignment, probably low standard deviation here. And so this would be, could be considered a good place to stop and take a recording. But for the purposes of trying to be confident in what we're doing, we continue to turn, and then you can see that when you go even more peripheral, you get even more hyperopia. You start to get misalignment of the eye, so it's not really on axis, and then the pixel brightness and the image brightness doesn't really change. And so this sort of saturating part of this brightness curve is where we would stop and try to take it. And this is rotating in one direction, but we actually, to verify, sort of do an internal verification of any measurements that we get, we also go in multiple directions. So we would go back to the optic nerve at this point, and then we go in the other direction. And you can see a very similar trend is happening here, where we get to some point and the alignment is good and the pixel brightness is good. But if you keep going, you don't really get any better. And so we usually go in both horizontal directions, and then as a check, we will also go in the vertical directions. And this is done by carefully turning, rotating the head towards or away from the camera. And so you can see as you do that, you get similar you start to get this same type of hyperopic refractions, at least in this example. And so based off of this, we would then find sort of the best fit. We know around where it is, where we should be taking the measurement, and so then we make sure that all the parameters and thresholds are met, and then this is where we will take our recording. And so for anesthetized recordings, we have a lot more control over the system, and so we require 20 stable measurements that have more strict criteria. So a standard deviation has to be below 0.5 diopters. The gaze has to be below 5 degrees, and often both of these measurements are even less than this usually. But this is the sort of the threshold. But we still keep this this criteria that the two eyes need to match within 2 diopters to be able to include this animal. Okay, so now I want to show some data from a variety of studies plotting this refractive error data, so we can see how it looks. And so here is from one of the original papers. This is refraction on the y-axis over age of the mouse on the x-axis. And you can see that at earlier ages, at younger mice, they start off slightly hyperopic kind of at baseline, and then they get a little bit more hyperopic over time and then saturate. These experiments here were performed in awake animals. And this seems to be a sort of a typical C57 black 6 refractive development curve. But there is this, idea about how does awake and anesthetized refractions differ. And so this was studied a little bit further in a study from the Purdue laboratory. And so here is plotting asleep refractions on the y-axis and awake refractions on the x-axis in the same animals. And you might be able to appreciate that they match pretty well, but there's a really large difference in the error, the variability here. And this was further documented in a more recent study where they compared awake measurements over time with measurements under anesthesia. So you can see in the top part of this figure, the awake measurements are really quite variable in that was coming out of this laboratory, but this was significantly reduced under anesthesia. And so the conclusion here is that asleep refractive measurements are clearly more stable than awake, but they can also be used to verify awake measurements in a more precise manner. Okay, so at this point, a number of laboratories have used the photorefractor to monitor refractive development, and there is some non-uniformity in these measurements across studies. So here is a table that just highlights a number of different studies, the instrument that was used and the refractive error in C57 mice. And if the red box, I've highlighted the studies using the photorefractor. So if you just kind of look at the refractive error measures, you can see that several studies find this sort of hyperopic, this positive refractive error at baseline, but several studies also see that it's closer to 0, actually, no refractive error. And it's unclear the cause of these differences, but I've highlighted some here that are important to consider. One could be the refraction technique, right? How you're orienting the eye and what part of the fundal area you're using to measure, and whether this is consistent between cohorts of mice, for example. Another is camera settings. So camera settings can change brightness levels, and so maybe this is different between studies. Anesthesia possibly could be playing a role here. There is some evidence that anesthesia may change refractive error. But a number of laboratories also do not see this difference, so it's unclear what the cause is for that. And then also restraining techniques during awake refractions may also be in play here. But I do want to point out again that despite some of this variability, there is a lot of consistency between studies. So here is a graph showing refractive error over age from a number of different studies. So now this is over time, unlike the table. And you can see that many of the studies sort of in C57 mice start kind of hyperopic in their refractions, and then they get a little bit more hyperopic over time and kind of saturate. But a couple studies here actually show myopic or less hyperopic refractive error. Okay, so the photorefractor enables us to measure and monitor refractive error over development, which is great for being able to understand kind of this idea of how refractions change, but we want to now be able to apply it to a myopic state. And myopia can be induced in a number of animal models. I'll just be talking about the mice for today. And the way that myopia is induced is usually two methods. One is form deprivation, where a diffusing lens, a frosted diffusing lens, is placed over one eye of the animal, and so that eye receives kind of a blurred image. Another technique is to use lens defocus. So a minus diopter lens is placed over one eye of the animal, and this changes the focal point of the light that's entering the eye and causing a blur. And usually either of these options are done at earlier ages, 3 to 4 weeks of age, so 21 to 29 days typically. And typically 2 to 4 weeks of lens treatment is sufficient to induce myopia. In the mouse, of course, some studies will take this out longer. And so the first use of this was from Schafel 2004 paper. And so here you can see an image of the mouse. Right on its right eye there, you see the frosted diffuser lens. This is a form deprivation model. And in the graph is measuring refractive error over age. The open circles, the small open circles and the black circles show control animals or the eye that did not have the lens. And you can see they start hyperopic and they get slightly more hyperopic over time. However, the deprived eye that had the lens actually has a pretty flat refractive development curve. And so this eye is relatively myopic. A few years later, another iteration of this procedure, this lens treatment procedure, was created. And you can see in this image of the mouse here, this is more like a monocle lens that's fitted over the one mouse eye, and then it's fixed to the mouse. But we see the same sort of results here. So the three development curves at the top of the graph are all control in either a control animal or in the eye that was not had a form that was not form deprived, and they start sort of hyperopic and get more hyperopic over time. But the eye that was form deprived kind of has this a stable refractive development that actually gets a little bit more myopic. And then I just want to point out more recently yet another iteration of this induction method that's similar to the previous one was created. And here you can see the example and image here, but in this particular image, they are the authors are using a minus diopter lens and minus 10 lens, which is what's shown in the graph. And this is showing the change in refraction. So when an eye did not have a negative powered lens, which is the blue line, they get more hyperopic over time, whereas the lens defocused eye gets more myopic over time. Okay, so we have the ability to measure refractive error, so that's reliable, and we also can induce myopia in mice. So the Pardue lab is very interested in understanding and kind of asking this question: what signaling pathways are involved in myopia progression? We know the visual environment regulates myopia, and there are many aspects of the environment that regulates myopia. So data I will be showing from previous studies in the Pardue lab will incorporate some of these visual environment changes. But if the visual environment is important in myopogenesis, then it must be signaled by something in the retina. So retinal signaling pathways may also be involved. So here is a schematic of, just like a cartoon schematic of a retina, showing photoreceptors at the top. They will, of course, activate internuclear cells, which then activate ganglion cells. Of particular interest, because of this aspect of ambient brightness that's important in myopia, are the rod and cone pathway systems. So rods, which are noted in black here, these are active under dim light conditions, whereas cone pathways are active under bright light conditions. There's also a third photoreceptor type that are intrinsically photosensitive ganglion cells. I have this shown here in blue, specifically the melanopsin retinal ganglion cell. The Pardue lab has also looked at these. And then lastly, dopamine is been demonstrated to be involved in myopia and is considered protective. And so we also have looked at any involvement of dopaminergic signaling in myopia. And so the question has been, the main question in the lab is, how do these different light properties, photoreceptor pathways, and then neuromodulators affect refractive development and myopia? And so I want to just show a series of results, refractive data from several studies from the Pardue laboratory. And the first is involves wavelength of light. So we know that indoor environments are deficient in short wavelength light, such as violet light, for example. And so in this particular experiment, mice were housed in different wavelengths of light during this refractive development period. And so here is showing refractive error over time. And the black line here is when the mice were raised in white light, and then green for green light, and then this violet color for violet light. And so in both white and green light, mice showed a very typical sort of hyperopic refractive development curve. But this was even more pronounced when they were raised in violet light. So violet light seemed to make the eyes even more hyperopic. And then cohorts of mice were made myopic using lens defocus, a minus 10 diopter lens. And so I can walk you through these ones. In the top, in panel A here, shows white light. So you can see in the contralateral eye, this is the eye that did not have the lens treatment. The animal showed a very typical hyperopic development curve. This is a solid black line. But in the lens treated eye in the dashed line, there was significant myopia development. When the mice were raised in green light and this treatment was performed, interestingly, there was less myopia development. So you can see that dashed line doesn't quite reach the same negative refractive errors, but it kind of remains pretty flat. And this was even more pronounced in violet light, where for at least a couple measurement time points, both the lens treated and the untreated eyes followed the same development curve, and then the lens treatment eye sort of saturated here. So there's even less myopia development, or less myopic refractive errors measured. And in panel D shows what you'll often see in a number of studies, which is plotting the refractive shift or the myopic shift, which is just taking the difference between the two eyes. So a more negative value means that more myopia was induced. And you can see that there's a significantly less myopia that was induced in violet light than in white light. Okay, so this kind of let us know about wavelength, but another important aspect of ambient environments is the brightness level. And so we know bright light is protective against myopia, but it was also found in the Pardue lab in another study that less myopic children also tend to spend more time in dim environments. And so this was really interesting. And so we, in the lab, tested this kind of idea by housing mice under different ambient light levels during refractive development, including very dim light. And so here is refractive error over age. The black line is scotopic environments. These are the very dim environments where only rod pathways would be active. The orange line is photopic, bright, sort of daytime environment. And the blue line is this intermediate environment that is often, present indoors or in low light conditions. And it appeared that ambient brightness didn't really have any effect on refractive error measurements just over sort of a typical development. And the mice showed this also typical kind of development curve. However, there was something interesting that occurred after myopia was induced with lens defocus. And so here is plotting the myopic shift again. So remember, the more negative value here means there was more myopic, the animals were in a more myopic refractive state. And interestingly, both photopic, that bright condition, and scotopic, the dim condition, showed less of a myopic shift than the mesopic sort of intermediate light levels. And so it appears, based off of this study in mice, that bright and dim light produce less myopic refractions. Okay, so we know from this kind of work that ambient light and wavelength are affecting refractive development in myopia and can be measured nicely with the photorefractor. And so the next sort of question is, okay, well, how are these sort of environments translated into what's going on with the signaling pathways? And so the Pardue laboratory has also done a lot of work on trying to understand the contribution of different photoreceptor pathways to myopia. And so one study looked at cone photoreceptors, with the idea being that if bright light is protective, then cone photoreceptors may be important in this protection. And so cone photoreceptor function was disrupted using a GNAT2 knockout mouse. So now the following studies will really highlight one of the benefits of pursuing these types of questions in the mouse model, which is the genetic manipulations. And this manipulation alters some of the signaling function of the cone. And so here is refractive development curves. There didn't seem to be any difference between when cone photoreceptors were functional, which is in black, the wild type mice, versus the knockout mice in red. And after form deprivation, the wild type mice became more myopic, which is the black line. And then the knockout mice, the cone photoreceptor knockout mice, also became more myopic. And then after several weeks, they started to become even more myopic. But this occurred a little bit later. And so from this study, one conclusion is that the cone photoreceptor dysfunction just has a modest effect on induced myopia and no effect on refractive development. And so if cone photoreceptors only have a modest effect on refractive development and dim light may be protective, then perhaps also rod photoreceptors are important here. And so this was another photoreceptor pathway that was tested in a GNAT1 knockout mouse, which is very similar to the GNAT2, but just a rod-specific version. And here is showing refractive error development. The control, like wild type animals, had a very typical refractive development shown in black. But really interestingly, the knockout animals, the rod photoreceptor dysfunctional animals, shown in red, at baseline were already very hyperopic. So, and this hyperopia sort of was maintained over development. And really intriguingly was the difference that happened with induced myopia. So form deprivation was used in these experiments in control animals, which is panel A, or wild type animals, which is panel A. There was this very typical myopic shift occurring, and this was completely absent in the GNAT1 knockout animals. So, and this was really pronounced, as you can see here. And so it's clear that rod photoreceptors, based off of this study, are necessary for the response to form deprivation and for induced myopia in the mouse. And then besides rods and cones, I mentioned there's this other photoreceptor type, which are these intrinsically photosensitive ganglion cells. And expression of OPN4, which is the gene that encodes melanopsin, which is the opsin that makes these ganglion cells photosensitive, or intrinsically photosensitive, was shown to be altered in a chicken model of myopia. And so this was also a pathway that was investigated in the Purdue laboratory. And the MRGC, which is the melanopsin ganglion cells, their function was disrupted two ways. One, in an OPN4 knockout mouse, so these mice did not have this opsin, so they could not intrinsically respond. And then another mouse mutant had these cells just completely ablated, which is the OPN4 DTA mouse. And so there were some quite drastic differences in refractive development between these different mutants. The black line here shows the wild type animals showing a typical curve. When OPN4 was knocked out, which is red, these animals were significantly more myopic at baseline, but then actually became more hyperopic. So they're at later ages. So their development curve is very different. And then the DTA mice were just more hyperopic overall. So modulation of melanopsin signaling really has affected here refractive development. When these animals were form deprived, we can see that both of the OPN4 knockout animals developed more myopia than the wild type animals, as shown here, and they were not really different from each other. So in this particular study, we conclude that melanopsin is necessary for refractive development and is involved in myopia. And so the last study I'll show is looking at dopamine signaling. And so dopamine has been really well studied across many animal models and is demonstrated to be involved in myopia development and is considered a stop signal for myopic eye growth. So the question here was testing dopamine deficiency and how does this affect refractive development? And this was using this retinal tyrosine hydroxylase knockout mouse. And so this will should make the retinas deficient in dopamine. So here's refractive development. Wild type control animals in black showed a typical curve, and this was slightly reduced, and knockout animals had slightly reduced hyperopia in these when they were dopamine deficient. But interestingly, with form deprivation, there didn't seem to be a difference between the two animals. And so dopamine signaling is clearly important for refractive development, but maybe not so much in form deprivation induction in mouse myopia. Okay, so in summary, the work in the Purdue laboratory has found that multiple visual pathways affect refractive development in myopia. For the visual environment, it appears that dim and bright light are both protective against myopia, but this sort of intermediate light is not. It appears that shorter wavelength light are more protective than sort of white light. And then in terms of retinal signaling, rod pathway systems and these intrinsically photosensitive systems seem to have a lot more modulation of refractive development in myopia than perhaps cone pathways and in some cases dopaminergic pathways. And so the takeaway points from this is that myopia is a critical health problem worldwide, and we really need to understand the mechanisms that underlie its progression. The photorefractor is a critical part in working towards this goal because it actually allows us to monitor refractive error in the mouse model, which has traditionally been difficult to do with the size of the eyes. And then different types of manipulations of mouse models can be precisely quantified with this photorefractor. And then finally, work in the Purdue laboratory and still ongoing work that's interested in continuing these studies has found that really multiple signaling pathways affect refractive development in myopia further complicating this disease. But we're working towards a better understanding with each of subsequent study. Great, and so with that, I would like to acknowledge everybody in the laboratory. Dr. Michelle Pardue, of course, she's, her career has been focused on trying to understand these challenging sort of questions in the myopia field, and then also, developed a lot of some of our techniques very early on with the mouse model and the photorefractor. And then also current laboratory members that all are using this device as well. I'd like to do a special shout out to the previous lab members whose work I just talked about, and everybody's listed there. I'd like to thank the funding as well, and then Striatech again for allowing us to be able to talk about some of this work and how we use the photorefractor. So thank you.
Thank you, Reece. Very interesting, and we already have a lot of questions pouring in from the Q&A. So if you don't mind, Reece, I would just dive right into the Q&A.
Okay. Do you need to unshare my screen or?
No, just leave it as it is. Yeah. So first question that came in was how you actually reduce variability when taking the measurements, other than putting the animals to sleep.
Yeah, so this is a good question, and this is key, I think, when using the photorefractor, because there can be a lot of variability here. So one thing is to have sort of for inclusion criteria, to have some inclusion criteria here, right? So we only will take like refractive error measurements that are less than 0.5 diopters standard deviation, for example. So having some inclusion criteria can help to reduce variability. It may require running more animals. The other component here is really working towards the alignment issue, or the alignment challenge rather. So the photorefractor software has this kind of alignment that tells you kind of an alignment, gives you an alignment measure. But trying to always, how we do it is we always will start with focusing on the optic nerve, and then we will sort of rotate the animal so that we're not on the optic nerve anymore. And then just enough so that the brightness is sort of its peak brightness. And this, I think, helps to keep the location on the fundus sort of similar across animals. So this is another way to help here. And then finally, trying to make sure that the eye looks really good. So if there is some dryness of the eye or anything like this, tear film issues, this creates a lot of optical errors and aberrations, which make refracting the animal very difficult and interferes heavily with the measurements. So those are some ways that we try to minimize sort of variability when we're taking the measurements.
Do you use any eye drops or artificial tears during the experiments?
Yeah, so this is a really great question. So we do not do that during the experiment. So if so the kind of order of events is we'll dilate the eyes, and then we will check. So we'll have the animal, like we're doing an awake refraction, we'll check the eyes to make sure that it's dilating okay, it looks really good. At that point, if you notice something, then you can apply a drop or refresh tears or something like this, and then give the animal the opportunity to kind of blink and clean the eye. And then during awake refractions, if you notice this, you also have this ability. Once you put the animal under anesthesia to do sleep refractions, I would suggest not to do anything to the eye. So it's sort of, you're at the mercy of kind of the eye at that point. We have found that if you try to do that while they're under anesthesia, it's just very difficult to kind of wick away all the excess liquid to get a good sort of uniform kind of eye layer. And then yeah, we just haven't, we tend to not want to touch anything with the eye once they're asleep. But for awake refractions, yes, you can put some saline or some refresh tears or something. Yeah.
Mm-hmm. All right, thank you. Given the variability of the measurements, if you compare two groups of animals, how big of a refractive difference do you need between the groups to be able to resolve it? I mean, that they actually are different, and what group size do you need in order to be able to do that?
Yeah, great. So the first part of the question, and an important one as well, is that when we measure like myopia, for example, we always will put a lens on one eye and then the other eye is not really treated with like a visual disruption. And this allows us to calculate like a myopic shift. While we have those animals, we also include naive animals. So these are animals that both eyes are undisrupted. And so we can compare the two. So a lot of the graphs I show, if you remember, you will notice that there's this sort of control animal, and then there's an animal that gets myopia. And so this allows us to see if this difference is sort of real or not. If the control animals also see there's a lot of variability in between their two eyes and between the measurements taken, then we're going to get less sort of significance. So this is one way that we actually are able to know, even with smaller amounts of change in their refractive error, that there is actually a difference. But that being said, there is a lot of variability and sometimes in these measurements, and a change of maybe one diopter, unless you're comparing it to a naive animal and you have many animals, you may not be so confident that this is actually different. So several diopters of difference is usually what we get. That seems to be a significant difference. And then this requires a few more animals. So at least 8 to 10 animals usually is what we'll need based off of experience to kind of get a good spread of data, because mice can have slightly different refractive errors, even just between mice.
Mm-hmm. Yeah, you mentioned that when you listed the quality criteria of what animals to include in the study, that you want to avoid animals that have a diopter difference between the two eyes of more than 2 diopters.
Yes.
What happens to that criterion in animals that you treat monocularly with a lens? You just don't apply that?
Yeah. So this criteria is for baseline measurements. So sorry if that was unclear. So before we treat any animals with any sort of visual disruption, lenses, et cetera, we'll do baseline measurements. We usually will do these at 28 days of age. And so at baseline, if the two eyes are more than a couple diopters difference, we will exclude those animals. Sometimes this happens. It's not very often, but this can happen where you can get, one eye is 8 diopters and another eye is 4 diopters or something. And depending on which eye is which, the eye that we want to apply the lens to may already be myopic. So we would exclude an animal like that. Even sometimes more rarely, you can get a hyperopic and a myopic eye. And when we see stuff like that, maybe something else is going on in this animal. And so we exclude that. But this is only done at the baseline measure.
So you nicely explained that anesthesia reduces the variability in the measurements, but could it have other effects on the measurement that we have to take into account?
Yeah, so right, exactly. So we in our laboratory, we do not really find an effect of anesthesia, the typical sort of ketamine, xylazine anesthesia that's often used. We don't really see an effect with that on refractive error measurements like between awake and asleep refractance. But there is evidence that anesthesia could have an effect. One study out of the Tuchenko laboratory found that in awake animals, actually, they were measuring closer to around 0 diopters of refractive error, and even adding some tropicamide caused a slight hyperopic shift, and then ketamine xylazine caused an even more hyperopic shift to around, like, I think 5 diopters or something, similar to what many people are measuring. But this was specific for this type of anesthesia. They found that a different type did not induce this. Like I said, we haven't noticed that this is occurring between our awake and sleep refractions, and our refractions are match others that also measure in awake animals. So I'm not really sure what the discrepancy is for this. It could be some other techniques happening possibly, but it's something to keep in mind as well.
Mm-hmm. Okay. Then there was a question that says, by defocusing the light, we also reduce its intensity. Shouldn't a conservative interpretation of those classic experiments be that the eyes of those mice are myopic because of reduced light exposure rather than the lack of a focused image on the retina?
Yeah, that's an interesting question. So some of the lenses reduce light, but some of the certain lenses, like some of the minus lenses, may not reduce light so much. But sure, if you have a really big, you use a really heavily frosted goggle, or excuse me, a heavily frosted lens, and there's some light reduction, this could be adding to this myopic phenotype. I will say that from our laboratory, when we have house mice in dim light, we don't see as much myopia, and so presumably these animals also have the lenses on that's reducing light even more. But yeah, there's certainly something interesting to think about here. But I'm not sure exactly how much, or I cannot recall rather, how much the light is actually modulated through these lenses, but certainly this would be the case. Another component of this that can add on to this idea is that most or all of the lenses also filter out short wavelength light. And so this might also be another sort of component to this that should be considered. Yeah, thanks.
Mm-hmm. Next question, are any mouse strains harder to measure than others?
The short answer is yes on this. Generally, C57 B6 mice are fairly, as far as easy or hard, I would put them on the sort of easier side to get data from. But because there's a lot that can change with certain mutant models, there can be other factors that sort of make using the photorefractor a bit challenging. One of these can be like dryness of the eye. So in some mutants that we've tested, we've noticed that the eyes seem to be dry and there's like some tear film kind of issues here. And this makes it very challenging sometimes to get sort of refractive errors that you're confident in. And so one workaround with that, which I mentioned a little bit ago, is that you can try to do things like before you measure to hydrate or kind of fix those issues, such as using artificial tears or saline or something. Also, the tropicamide, dilation drops may be slightly irritating, and so in some of these animals where it was more difficult, after we dilated, we would also try to sort of flush the eye with some artificial tears or something, and then let the animals kind of blink and stuff. And this actually seemed to help quite a bit. Another important component of this, which is more relevant for asleep refractions, is the development of cataracts. So sometimes these can develop for unknown reasons, or maybe it's related to anesthesia or something. But when this happens, you also kind of are out of luck a little bit with getting some precise refractive measurements. So these are some things to keep in mind, and maybe certain mouse strains are more predisposed to getting some of these types of things.
All right, and there's one question left, at least for now, and that is, what would the pathway be that carries a signal from the retina to the sclera? I don't think any direct retina sclera projection has been shown. So which brain center would this pathway go through? Melanopsin RGC target regions, but have they been shown to send projections back to the sclera?
Yeah, this is a great question. This is something that we are like actively interested in pursuing. Again, unfortunately, we don't really know what the pathway is. We know that multiple sort of retinal pathways are involved, and I showed some of the data from previous work in the Purdue lab. And it's possible that this includes these melanopsin retinal ganglion cells. I don't know if anybody is aware of projections from any of the regions, the brain regions that sort of come back to the sclera. Kind of more prevailing thoughts in the field currently is that there is a translation of some retinal signal that then like crosses over into the, crosses the choroid and then gets to the sclera. What this signaling molecule is or molecules rather, is still unclear. Dopamine is likely involved here. All trans retinoic acid and retinoic acid signaling is also probably involved. There's been a number of recent studies that have looked at this. And so it seems like this is more likely than necessarily visual information sort of leaving the eye to the brain and then coming back. I will say there are connections between the melanopsin retinal ganglion cells and dopaminergic amacrine cells. So there are some connections there locally within the retina. So if these cells are part of the pathway that's kind of controlling this, they may be able to influence dopamine signaling at that level.
Okay, I think we're right on time. So Reece, thank you again so much for showing us all that data and also the methodological aspects of doing those measurements. I think that is very interesting for many of the people that use that equipment.
Yeah, great. I'm happy.
Yeah, that was great. And also I want to thank the audience members for joining us and also for the great questions in the Q&A. I wish everybody a great day and invite you to our next journal club, which will come up in a couple of weeks. So thanks again to everybody and bye-bye.